So, welcome to the immunology lectures, and in this lecture we are going to discuss mainly about the regulation of the complement system. So, in our previous classes about the complement system, we had discussed about the complement activation pathway. So, how the complement is being activated, how the complement pathway is being activated. And in this complement activation pathway, we have learned that at least 3 different pathways are active, the classical, the alternative and the lectin pathways. And if we see that the major part of this complement activation pathway, or the central part of this complement activation pathway, is breakdown of the C3. So, I told in my last lecture as well that these complement proteins, they are always present in our system, but they are normally not doing any harm to us. They are not killing our self cells or our own cells, the healthy cells. They are not acting on the healthy cells, or they are not killing our own cells. They are mostly active on the pathogens or the foreign cells. So, they can recognize the foreign cells, and the complement protein or the complement system, as we told previously as well, that it is the effector part of the humoral immunity. So, one of the major effector pathways of the humoral immunity, that is in conjugation with the antibodies, it can lead to the complement activation, leading to formation of the membrane attack complex, or it can lead to opsonization of the pathogens. So, but the complement system does not work on our own cells, it does not do any harm to us, although the proteins are there. So, why they do not get activated? Why? The question is why and how. So, of course, there is some tight regulation of this complement system, and there is a class of proteins, a group of proteins rather, which are known to function as regulators of complement. So, they work as the regulators of the complement, and they are known as the regulators of complement activation, or the RCA proteins. And these proteins are encoded on a single location in the human chromosome number 1, and they are produced from there, and they work on this complement pathway at different stages of the complement pathway. Now, if we quickly look into the complement activation pathway, what we have seen in the last lectures, what are the major significant steps in the complement activation pathway? One of the significant step is activation of the C1 complex, that is C1 forming C1q-R2S2, and then it gets activated and cleaves the C2 and the C4. And once this C2 and C4 are cleaved, then only they can form the corresponding cleaved product like the 4B, the 2A, these are formed, and then only this 4B 2A can associate. So, one of the important steps in the complement activation pathway is the initial cleavage of the C2 and the C4 by the C1, and for that you need the activation of the C1. So, one of the major steps where these complement regulators can act or they can inhibit. So, these regulators, you have to remember that these regulators are kind of inhibitors. That means, they inhibit certain steps of the complement activation pathway or regulate certain steps in the complement activation pathway. And how do they do that, and which steps do they really act on? So, which are the major steps? One, as I told, is the C1 activation and the subsequent cleavage of C2 and C4 into 2A 2B and 4A 4B. The second major step is formation of the C3 convertases. You see the C3 convertases. The C3 convertases, there are 2 major C3 convertases that are formed. One is the 4B2A and the second one is the 3BBB. So, these are the 2 major C3 convertases that are formed. So, the second stage of regulation is the formation of the C3 convertase. That is, do not allow the C3 convertase to form, does not allow the C3 convertase to be formed. A third stage of regulation would be if the C3 convertase is formed. Let us say the C3 convertase, the regulation at the initial stage is somehow did not work, and the C3 convertase is formed by association of 4B and 2A or 3B and BB. So, a third point of regulation is dissociation of this complex, that is destruction or decaying of the C3 convertases, so that the C3 convertase cannot work on the C3 and lead to subsequent cleavage of C3 into 3A and 3B. Another point of regulation is, one of the major points of regulation is the regulation of formation of the membrane attack complex, that is this part, the last part. So, here, so another point of regulation is do not allow, although you can have all these C3 convertase, C5 convertase, but do not allow the formation of the membrane attack complex, so that the membrane attack complex is not formed. And a very tight control at the, you can understand a very tight control is required at this level of formation of the membrane attack complex, because sometimes, not all the times, usually as I told that the complement proteins, they assemble and activate each other on the surface, on the cell surface of the pathogen. But sometimes these complexes can also occur not on the cell surfaces. So, they can just occur outside the cell, and these complexes, if they form, and they can form a C5b6-7 complex as we described. So, if that kind of a complex is already formed, then that complex is competent enough to go and get inserted into a membrane and cause a hole or a pore or a channel in the membrane, leading to the lysis of an innocent cell. That means a cell which is not destined to lyse, which does not is not required to lyse. That kind of an innocent cell can be also lysed, and that is also known as innocent bystander lysis. So, to avoid innocent bystander lysis, you can also have regulations or checkpoints at the level of formation of membrane attack complexes. So, we see at least there are 1, 2, 3, here 3 and here 4. So, at least 4 different areas or 4 different parts of the complement pathway where you can have checkpoints or regulations. Now, let us have a closer look into this regulatory pathways, how the complement system is actually regulated. So, we start from the very beginning in the classical pathway, where we have this C1q R2 S2, and there is, so this, as I told or described them as regulators of complement activation. So, who are they and how they work? So, one of the very initial stages where these RCA proteins can work is on the C1 complement protein, and one of the important RCA is the C1 INH or the C1 inhibitor. Now, this C1 inhibitor, it binds to the C1 and inhibits the activation of the C1. So, what is the C1 or the C1qR2S2 known to do? It is known to cleave the C2 and the C4 and produce C2A, 2B and C from C4, C4A and C4B. And then this 4B and 2A they associate to form C4b2A, which is also the same from the lectin pathway. So, the first step of inhibition is the C1 INH or the C1 inhibitor, also known as the C1 inhibitor, which basically inhibits the activation of the C1 and inhibits this step of cleavage of C2 and C4 into C2A2B or 4A4B, and thereby this can inhibit the subsequent steps of formation of C4B2A as well. So, this, as we know, is known as a C3 convertase. So, this is one of the initial checkpoints where you can actually inhibit the formation of a C3 convertase. Now, what is the C3 convertase in the alternative pathway? We know it is the C3bBB. How does it come from? It comes from the C3b. Here is the C3b, and this C3b can associate C3b, which can associate with factor B and factor D, and lead to the formation of C3bBB. Okay. So, this is another C3 convertase. Now, as I told, apart from inhibition at the level of C1, that is at the beginning, there is also regulation at the stage of formation of the C3 convertase. So, there are regulators which does not allow the formation of the C3 convertase, and what are they? So, one of such regulators is C4b binding protein. As the name suggests, it can bind to the C4b. So, one of them is C4b binding protein, or there is also MCP, the membrane cofactor protein, or you have the complement receptor 1 or the CR1. They all can bind to the C4b and does not allow it. So, it inhibits its association with C2A. So, now this C4b, which was supposed to associate with C2A to form C4b2A, cannot associate with the C2A anymore, and thus these proteins C4bBP or MCP or CR1, they can inhibit the association of C4b with C2A. So, it inhibits this particular process. Similar to this, what we have seen in case of the classical pathway, similar to this in the alternative pathway as well. There are similar inhibitors which inhibits the step of association of the C3b. So, for example, the CR1 or the MCP, these are the common factors, these are the common regulators. Along with that, there is another factor H. This also inhibits association of C3b with factor B. So, the C3b now cannot associate with factor B, and if it cannot associate with factor B, then it cannot form the C3bBB. So, this cleavage does not occur, that means the factor B cannot be cleaved into factor BB, and this C3bB C3 convertase is not formed. So, these are the two major steps of regulation where the C3 convertases are not formed. Apart from, so in this same stage of regulation, when this C4b binding protein or the MCP or the CR1 they can bind to this C4b, they also activates another factor known as the factor I. The factor I is interesting. What does it do? So, this factor I, it basically it cleaves the factor 4. So, this is for example C4b, and this C4b is further cleaved into the C4c and the C4d. So, C4b, which was the membrane bound form, it can now get cleaved into the C4c and the C4d. The C4d still remains bound to the membrane, while the C4c is the soluble part which leaves or goes away after the cleavage, it goes away. Thus, it leaves the C4b inactive. So, the factor I is a factor which is primarily activated after binding of C4b-BP or the MCP or the CR1 to the 4b and leading to cleavage of the C4b. So, the C4b is then cleaved into 4d and 4c, where the 4d is the bound part, 4c is the soluble part, and this soluble part goes away. So, leaving out that the factor B is quite inactive and it cannot further form the C3 convertase. Similar to this, in case of in the case of C3 as well, this C3b here, this C3b here can also be further cleaved by the factor I. So, the factor I here cleaves the C3b. So, the C3b is also membrane bound form, we have seen previously, it can get bound to the membrane. So, C3b, which is initially in the membrane bound form, now it gets cleaved into so this is the C3F and this is the IC3B. So, the IC3B is the insoluble fraction. So, it still remains bound to the membrane, and the C3F is a small fraction, that is the soluble part, that gets cleaved and goes out. So, the C3B is broken down into the IC3B and the C3F. Now, this IC3B can be further broken down, and it can be broken down into a soluble and an insoluble fragment. So, we have the C3C and the C3DG. Now, this DG part remains bound to the membrane, while the C3C is the soluble part. So, now this C3B by the factor I it can be cleaved by this factor I, similar to what we have seen in case of the C4b. This C3b can also get cleaved by the factor I into IC3b and the C3F, which the IC3b can further be cleaved into C3DG and the C3C, and this cleavage leads to inactivation of the C3b. So, the C3b, as we have learned from our previous lectures, that the C3b is one of the central mediators of the complement activation pathway. It is one of the central molecules in the complement activation pathway. Now, this C3b is completely destroyed or degraded, and once this C3b is completely degraded or destroyed, it cannot further associate with the factor B or the factor D and lead to the formation of the C3bb. Now, this C3bb or the 4b2a, either of these two C3 convertases they converge at the formation of two different C5 convertases. So, they forms the C5 convertases, the C4b2A forms C4b2A3b, and this forms C3b BB C3b. Okay. So, these are the two C5 convertases. So, now another checkpoint in this whole activation pathway is decaying or dissociating the C3 convertases by itself, and that is done usually by a factor known as the decay accelerating factor, or the DAF, the decay accelerating factor. And this decay accelerating factor also works on the DAF, can also work on the C3b BB convertase, that is the convertase or the C3 convertase from the alternative pathway. So, this DAF, the decay accelerating factor, is active on the C3 convertases. So, they work on the C3 convertases and they can quickly or very fast they can dissociate the 2A from the 4B or the BB from the 3B. So, dissociation of this 3B from the BB ensures that the C3 convertase is not active anymore. Similarly, dissociation of 2A from the 4B ensures there is no further C3 convertase available to convert C3 or to decay or destroy cleave C3. So, this is another point of regulation where we have the decay accelerating factor or the DAF. So, now once we have kind of seen the checkpoints starting from the C1 to the C4, C2, C3, and mainly the C1, one of the checkpoints is the C1, this initial part, and the second checkpoint is in the formation of the C3 convertase, and the third checkpoint is the stability of the C3 convertases. That means, the C3 convertases are decayed. Now, once the C3 convertase, if they skip these checkpoints and there is formation of a C5 convertase. So, what do? They cleave the C5, and the C5 is cleaved into 5A and 5B, C5A and C5B. So, it is cleaved into these two molecules or the two fragments, and C5B associates with C6, C7 to form the C5B67 complex. Now, this C5B67 is a complex which is already competent to go and lead to lysis of the cells. C5B67 can already, because the C7, the outer part of the C7, it undergoes a structural transition and leads to exposure of the hydrophobic patches. So, it can very easily go into the membrane and can lead to lysis of the cells. Now, if the complexes are 5B67 complexes are formed on the surface of the cell, there is no problem, or the target cell, there is no problem. But if it is not formed on the surface of the target cell, then what happens? If it is, it can travel to the next cell and can kill an innocent cell. So, there can be innocent bystander lysis. So, the next checkpoint or the point of regulation is how to stop this kind of bystander lysis. So, there are complement regulators or complement proteins, regulator proteins, which can bind to this. So, which can bind to this C5B67 complex and inhibit its insertion into the cell membrane. So, one of them is, for example, the soluble serum protein or the S protein that can bind to C5b67 complex and inhibit its insertion into the membrane. Now, even if it skips this checkpoint, you still have the C8 binding to it and forming C5b678. So, C5b678, and finally, you have C9 or poly C9 binding leading to C5b6789, which is actually the membrane attack complex or the MAC. So, this is the MAC. So, now there is one final more checkpoint where there are two major proteins. One is known as the homologous restriction factor or the HRF, another is MIRL, also it is also known as the membrane inhibitor of reactive lysis. So, membrane inhibitor of reactive lysis. So, now this MIRL or the HRF, they can bind to this complex, the C5b67 complex, bind to the C5b678 complex and inhibits binding of the C9. So, this inhibits the binding of the C9. So, this MIRL or the HRF, the homologous restriction factor, or the MIRL, that is the membrane inhibitor of the reactive lysis, both of these two proteins, they binds to C5b678 complex and thereby inhibits the binding of the poly C9 and formation of the membrane attack complex. So, there is no formation of the membrane attack complex. So, these are mostly the different stages where these regulators of complement activations or the RCA proteins they act upon. So, quickly I go through or revise this whole thing. So, there are the different checkpoints. The first checkpoint is here at the level of C1. It inhibits the activation of the C1 and cleavage of the C2 and the C4 downstream. The second checkpoint usually is in the classical pathway, it is the binding to the C4b, there is a cleaved product of C4. So, C4b binding protein or the MCP or the CR1, they binds to C4b and inhibits its association with the C2a. So, that this C3 convertase, that is C4b2a, is not formed. Similarly, this is also part of the second checkpoint or the second regulation, that is the CR1 or the MCP, the membrane cofactor protein or the factor H, they bind to C3b and inhibits the association of C3b with factor B. So, that C3b cannot interact or cannot associate with the factor B, and the finally inhibition of C3b complex. Now, apart from doing this, both of them they initiate a third process, that is they activate this factor I, they activate the factor I. So, here and here as well, they activates the factor I, which can cleave the C4b, that is the cleaved product, that is the one of the main mediators or one of the main components of the C3 convertase. So, factor I can cleave C4b into C4c and C4d, so that there is no further C4b available for the formation of 4b2a. Similarly, in the alternative pathway, the factor I can cleave C3b into C3f and IC3b. IC3b is further cleaved into C3c and C3dg, and so by that the factor I can cleave or completely destroy this C3b and inhibits or does not allow the formation of this C3b-BB complex. But what if this C3b-BB complex has already been formed or the C4b-2a complex has already been formed? That has been taken care of this fourth step or the fourth checkpoint, which is the decay acceleration factor here and here in both the cases. The decay acceleration factor usually is responsible for quick decay of or dissociation of 4b from 2a or 3b from bb. So, once this C3 convertases like 4b2a or 3b-bb is formed, this decay acceleration factor can dissociate the 4b from 2a or the 3b from bb and thereby inhibiting the C3 convertase state. So, it does not allow the C3 convertase to be formed. Further to this, we have two more steps in the process of the membrane attack complex formation. One step involves immediately after formation of 5b. So, immediately after formation of the 5b, we have 5b6-7 complex, and this complex is a very important complex in a way that this 5b6-7 can get inserted into the cells and can lead to innocent bystander lysis. So, S protein, one of the soluble serum proteins, so the S protein, it can bind to this 5b6-7 complex and thereby inhibiting this complex to get inserted. And how does it do that? So, after this S protein leads to a hydrophilic transition. So, usually this C7 is hydrophobic, and that is what favors its insertion into the membrane. So, now this binding of this S protein leads to a hydrophilic transition, so that it cannot get accommodated in the hydrophobic environment of the membrane. So, it does not get inserted. So, this is another checkpoint number 5, and finally, in the last step you have the checkpoint where these two proteins, that is the homologous restriction factor, the HRF, or the MIRL, that is a membrane inhibitor of reactive lysis, these two factors, they can bind to the C5b678. 5b678 is the penultimate step of the formation of membrane attack complex. Now, these two proteins, they can they are expressed on the surface of the host cells, and they can bind to C5b678 and thereby inhibiting or restricting the binding of the C9 to this 5b678, and if C9 cannot bind to 5b678, it will not form this poly C9 and the membrane attack complex. So, the membrane attack complex will not be formed, and this is the sixth or the final regulatory steps by the complement regulators or the regulators of complement activation. So, in today's lecture we have learned briefly about the regulations that are there in the complement activation pathway, and how these regulators of complement activation, they regulate the different steps in the complement activation pathway, and thereby does not allow the complement proteins to just get activated and destroy our own cells or work on the self cells. So, that is all for today's lecture, and we have mostly completed or finished with the complement proteins, and I hope you have understood the complement proteins by far. You have understood the complement proteins. So, we will finish the complement chapter here, and you can go through any standard textbook which has been referred to in our website. You can consult any of the textbooks and read them, and I hope things get clearer to you, and thank you very much.